smears, we take a part of the aspirate into a tube containing 10% neutral buffered formalin, and this is how a fresh sample taken in the neutral buffered formalin will appear. Now this tube is then allowed to stand for 24 hours or overnight, and then a nice pellet settles down at the bottom of the tube. This is for the effusion sample, and in the next tube we can see a nice pellet at the bottom after 24 hours of fixation in the neutral buffered formalin. Now both of these samples will be centrifuged. So the centrifugation will be done in this desktop or tabletop centrifuge, and the speed will be 1500 revolutions per minute for 10 minutes or 3000 revolutions per minute for 5 minutes. So now the centrifugation is over. We will now take out the tubes. So the tubes have to be kept balanced in the centrifuge. So now we can see a nice pellet, even more nicer pellet has settled at the bottom of these tubes. So this is the effusion aspirate and this is the effusion sample, and a nice pellet can be seen. So now we will discard the supernatant. Gently we will discard the supernatant from each of these tubes. So now, as in the thrombin clot method, we will give one step of washing. This washing will be done using the phosphate buffered saline, and this PBS solution or the phosphate buffered saline solution will be added into the tube. So similarly, it will be added into the next sample. So this washing is basically done to remove the formalin residues so that they don't interfere with the staining later on. So now we have added phosphate buffered saline. Now these tubes will again go into the centrifuge, and a nice centrifugation will be done for 5 minutes. So in contrast to the thrombin clot method, we will do this washing only once in this sodium alginate technique. So now it will go into the centrifuge, and same speed centrifugation, 3000 revolutions per minute for 5 minutes will be done. Now this centrifugation is over. We are now taking out the tubes. So more clearer pellet. The supernatant is also clear enough. Same for this aspirate sample also. So now for the next step, so before the next step, the supernatant will be discarded. So for the next step, we require two solutions. As the name suggests, the name of this technique is the sodium alginate technique. So first solution is the 1% sodium alginate solution that has been prepared in the laboratory using the sodium alginate powder, and we prepare 50 ml of sodium alginate stock solution, 1%, and this can be used for a period of 3 months. This can be stored at room temperature. Similarly, calcium chloride, 1 molar calcium chloride solution is prepared, and this can also be stored at room temperature. So now we'll be adding 500 microliters of sodium alginate solution to each of these samples, the pellets that have settled at the bottom. So now we can add this 500 microliter by using either the auto pipettes, or this can be added using the partial pipettes, and it is almost equivalent to 10 drops. So 10 drops of the solution can be added. So right now we are using auto pipette, and it has been set at 500 microliters, and drop by drop along the wall of the tube this will be added. So same solution will be added to the next sample. So 500 microliters or 10 drops of 1% sodium alginate solution. So next step is we will mix it and we will give a spin and centrifuge these tubes at 3000 revolutions per minute for 5 minutes. So this is to ensure proper mixing of the sodium alginate with the pellet. So the basic principle in this technique is that the sodium alginate will transform into a gel when it is mixed with calcium ions. So in presence of calcium ions, it will turn itself into a gel. So we have to ensure that this sodium alginate mixes properly with the sample so as to ensure that our sample is present in that gel. Now the centrifugation is over. We are taking out the tubes. So there is adequate mixing of the sodium alginate with the cell sediment. So now we will be adding 1 molar calcium chloride solution, 500 microliters, which is equivalent to 10 drops or 0.5 ml. So this will be added to each of these tubes, and this should be added at the bottom of the tubes so as to ensure proper contact of the sodium alginate with the calcium chloride and entrapment of this cell sediment within the gel. So within a few seconds, a nice gel forms within the tube. We can see the sediment has been trapped inside this gel. So now same will be repeated for the second sample. And by the time we are done with the second sample, our gel will be formed. So now 0.5 ml, 500 microliters or roughly 10 drops of the solution, 1 molar calcium chloride solution. So make sure you are putting it at the bottom of the falcon tube. So mixing. So now a gel has formed. And now this gel with our cell sediment, it will be processed as a small tissue biopsy. You can see a gel has formed, so it is not moving now. So now we will take out these gels on these filter papers that have been labeled with the sample IDs. We will gently take out this gel onto the filter paper. You can see our sample, our cell sediment is entrapped within this gel. This will be done for the second sample now. So gently we will be taking out. So these are both the cell blocks that have been entrapped within the gels, the sodium alginate calcium chloride gel. Now these will be folded and will be processed as small tissue biopsies. These will be packed into the plastic tissue cassettes and will directly go for tissue processing. We have to take care not to put these cell blocks for formalin fixation, as addition in formalin may dissolve this gel. So these will be going directly for tissue processing now in the automated tissue processor or in the manual ones, as is the availability in your laboratories. So you can see that this whole process is over in just 20 minutes. Good morning, everyone. I will be demonstrating the plasma thrombin clot method or the plasma thrombin method for cell block preparation. So this is the setup that we will be using, and so this is the sample that we have taken in 10% neutral buffered formalin. After preparation of the adequate number of aspiration smears, we rinse the needle in 10% neutral buffered formalin solution that has been added to this falcon tube. Subsequently, the tube is then kept overnight, and it is the sample is then allowed to fix in neutral buffered formalin. And this in the next tubes, you can see a pellet has settled down at the bottom. So in the first tube, the blue cap, we can see the final aspirate pellet that has formed after overnight fixation in 10% neutral buffered formalin. And the tube with the red cap, this is a effusion fluid specimen wherein the pellet has formed at the bottom after overnight fixation. So now we have to centrifuge these pellets so that all the material that has been suspended in the supernatant that settles down at the bottom. So now we will centrifuge these tubes at 1500 RPM for 10 minutes, or we can centrifuge them at 3000 revolutions per minute for 5 minutes. So now centrifugation has to be done. So this is the centrifuge, desktop centrifuge, tabletop centrifuge. So now the centrifugation is over. So now we'll gently open the centrifuge lid. Wait for the motion to stop. Now we'll take out the tubes. You can see nice pellets have formed at the bottom of the tubes. So now we have to do the washing step. The supernatant has to be discarded. Similarly, for the next sample, the supernatant will be discarded. So for the next step, that is washing, we will add phosphate buffered saline. So now this will be added to the falcon tubes with the cell pellet at the bottom. These are being capped. So this washing step has to be done thrice. So now these tubes will go into the centrifuge, and we will again centrifuge them at 1500 revolutions per minute for 10 minutes or 3000 RPM for 5 minutes. So now the centrifugation step is over. The tubes will now be taken out. You can see there are nice pellets at the bottom. So now this washing step has to be repeated again. So this will be the second washing. Again the supernatant will be discarded. And phosphate buffered saline will be added to these pellets. Similarly to the next sample. So this phosphate buffered saline is being added. So after adding phosphate buffered saline, these tubes have to be centrifuged. Again at the same speed, that is 1500 RPM for 10 minutes or 3000 RPM for 5 minutes. So now the second centrifugation is over for the washing step. The tubes will be taken out. You can again see pellets have formed at the bottom. So now again the supernatant will be discarded. Similarly, for the second sample, the supernatant will be gently discarded. Now for the third washing step, we'll again add phosphate buffered saline. We need to mix this phosphate buffered saline well with this pellet so as to ensure that nice pellet will be formed in the next washing step. Next centrifugation, after centrifugation, nice pellet should be formed. So now these samples will be centrifuged. So this is our final third centrifugation for this washing step. Again same speed, 3000 rpm for 5 minutes or 1500 rpm for 10 minutes. So now the third centrifugation is over. So this completes our washing step. So now the supernatant will be discarded. And now we have to add plasma to these pellets. So this plasma is the pooled plasma that has been obtained from blood bank. So this is the plasma, pooled plasma sample. So now we will add 100 microliter of plasma to each of these pellets. So the basic principle in this technique is to add plasma and thrombin to the cell pellet so as to obtain a clot that will enmesh the material or the cells within it. So now we have added plasma. So now after adding plasma, we will add thrombin. So this is the thrombin vial. So initially this thrombin originally comes as a lyophilized powder, and we reconstitute it using 8 ml of distilled water, and now 50 microliter of this thrombin solution will be added to each of these tubes. So we have to go down and then add thrombin so that plasma and thrombin, they come in contact with each other and a clot will form within a few seconds. Similarly, for the next sample, 50 microliter of thrombin will be taken and added into the tube within the sample. Ensure proper mixing of thrombin and plasma, and we'll keep the tubes, and within a few seconds a clot will be formed. So now we'll take these tubes and we will show you the clot. So can you please tilt the tube? So now we can see that a nice clot has formed at the bottom. So same has formed at the bottom of the other tube also. So now we'll take these tubes and we'll dislodge this clot that has formed at the bottom using either the pipette tip or you can use a spatula, but you have to gently dislodge it and then process it as a small tissue biopsy. So now we have put it onto the moistened filter paper that we have labeled with the sample ID, and now it will be packed and processed as a small tissue biopsy. So this is being put in a plastic tissue cassette, which will then go for the tissue processing in the automated tissue processor. So same will be done for the second sample. We'll gently dislodge the clot that has formed at the bottom of the tube. Now this will be put onto the filter paper You can see a nice clot. This is from the effusion sample. So the previous one was from the aspirate, final aspirate. This is from the effusion sample. This has been labeled with the ID, sample ID, and now we will pack it. Put it in the cassette and it will be processed directly. So now there is no need for formalin fixation. It can directly go for further processing. So we can see now our cell blocks are ready to go into the tissue processor, and this entire process has taken around 25 to 30 minutes. Thank you.